Hey guys, so we're continuing on our meiosis and genetic diversity, and to what this lesson we're looking specifically at meiosis, or sometimes you'll hear it said meiosis because they like to pronounce all the pieces. You'll notice it's very similar to mitosis in terms of the basic process, but ultimately you can almost think of it as a double division. So we make our copies, and so meiosis 1 would be our first division, and meiosis 2 would be our second division. This is how we end up with our haploid or N organisms, our gametes. So meiosis is a process that creates haploid gamete cells in sexually reproducing diploid organisms. We get a daughter cell with half the number of chromosomes as the parent. So for example, in humans, our diploid is 2N, which is 46. Meiosis produces sperm and eggs that are haploid or N23. Okay. So typically there are 2 rounds of division with this, meiosis 1 and meiosis 2. So what's the difference between mitosis and meiosis? So meiosis is similar to mitosis, but there are some really key differences that make it work the way it does. Mitosis, of course, occurs in somatic cells, and there's only a single division, and we get 2 diploid daughter cells that are genetically identical. In meiosis, we're forming gametes or gametic cells, egg and sperm. We have 2 divisions, and we get 4 haploid daughter cells, each genetically unique. So they're all different. So 3 key events are found in meiosis. The first one that's really unique is called prophase 1, and it's in meiosis 1. And the reason this is so unique is it actually we have this synapsis and we have crossing over that's occurring. In metaphase 1, our tetrads, the homologous pairs, actually will line up. So instead of everybody lining up single file, they're going to line up in pairs. So we can take one pair one way and one half of the pair one way and one half of the pair the other way. And then we'll split them further in the next round. And in anaphase 1, those homologous pairs are actually going to separate from each other. So we end up with the pair on each side before we split the others. Okay. So we start off with interphase. Interphase is the same whether it's mitosis or meiosis. Cell goes through growth 1, synthesis, where the DNA is copied, and growth 2. Everything's ready to go. Then we move into meiosis 1, prophase 1. So synapsis is the idea where those homologous chromosome pair up and physically connect so they form a little set of 4. We call that a tetrad because we want them to stay together until the point that we intentionally separate them. The other thing that's really unique to meiosis in this phase is crossing over. So it occurs at what they call the chiasmata, and the DNA is actually exchanged between the homologous pairs. So always think of it as if these 2 pieces, if they would actually almost like hug each other, but they hug each other so hard that the pieces break off and swap. So every single chromatid that ultimately is going to be produced is going to be totally unique. So we started off, we had 2 that were identical orange and 2 that were identical this purpley color. But when we do crossing over, we're going to have one that is maybe you have one that's solid orange, and then you have another one that's orange with a little tip of purple. And you have a solid purple and a purple with a little teeny tiny tip of orange. So every single one of them is going to be unique and different. Then metaphase 1 is the next kind of unique different in mitosis, all 4 of these would have lined up single file across the metaphase plate. But in meiosis, you'll notice the tetrads line up along the metaphase plate. So that in anaphase, we pull one whole pair in each direction from the tetrad. We need to do this because we still have to make sure we have a copy of every single one of the different chromosomes for our gametes. So we have to have enough available and ready to go, and we have to make sure we have one of everything for the second division. So our sister chromatids are still attached to each other. We have just pulled the pairs apart. Then we have telophase 1 and cytokinesis. So the nuclei and cytoplasm have finished their division. And so we have 2 separate cells, but we're going to continue. So we have a haploid set of the chromosomes in each daughter cell. At this point, it's actually N, because we only have 1 set of them in each one. We're just going to split them again to make them separate. So meiosis 2, prophase 1, this time there's no crossing over. We only do that in prophase 1. But other than that, prophase is pretty much the same across the board. So the spindles are going to form. The spindles attach to the centromeres. The chromosomes line up at the metaphase plate. Notice this time they're single file, just like they were in mitosis. And because of that crossing over we had originally, notice how each one is unique. So we have our solid orange and we have our orange with a little bit of purple. We have our solid purple and we have our purple with a little bit of orange on it, right? Anaphase 2, see how we've now broken the sisters apart, just like we did in mitosis. So sister chromatids separate and move towards opposite poles. And then telophase 2 and cytokinesis, we finished the nuclear division for telophase 2. We finished the cell division for cytokinesis, and we end up with 4 haploid cells. We got a nucleus reappeared in each. Each one is genetically unique. You can actually see on those pieces of chromosomes how each combination is slightly different. So quick review. Early meiosis 1, we have our parent cells. They're 2N. Meiosis, then we divide them and we end up with 4 genetically unique daughter cells that are N. So how does this lead to genetic variation? Well, first of all, remember we have crossing over. So that's where we get that recombinant chromosome. That's the one I talk about where it's like they hugged each other so hard they broke off pieces and then they hung on to their friend, so they ended up with a new piece. The independent assortment of the chromosomes. So when they're splitting, it's not like they're splitting intentionally to try and keep groups together. Each chromosome is going to separate all totally on its own. They're randomly set up along that metaphase plate when they split. And they can orient. It could be maternal on one side, paternal on the other. It's totally independent, totally up to whatever happens. And then, of course, random fertilization. So if we had 4 options, 4 different unique gametes, and we have unique gametes for the other one, we don't know which ones are going to come together to actually make the individual. So that's what we mean when we say random fertilization. Any sperm can fertilize any egg. So meiosis followed by fertilization gives us that genetic diversity that the sexually reproducing organisms are so well known for. There's so much genetic variation. And why this is really important actually will take us a few units through. We will be talking about it a little bit as we go through the next several units, but it really plays that critical role in natural selection, because when we're selecting from what's already there. And so this variation gives us a lot more to choose from. The cellular process drives the interaction of all those little subcellular components. It's using free energy that we have to have for the growth and reproduction. So that stuff, those big ideas that we've been talking about with the components, the free energy, it's all going on. Okay, so hopefully that will help you out a little bit on meiosis, and I'll see you for the next lesson. Bye.